Welcome to Study Loop.
So in this problem, we are given with two reactions of two drugs with a protein, and we are given the initial concentration of the protein and both the drugs, and we are also given the equilibrium concentration for the drug protein complexes. And from all these values, we have to find out the value of Kc for each of the drug protein reaction, and from there we'll have to say which drug protein binding is more stronger than the other. So we will start by writing the reaction equation for the protein with for the protein with drug A. So since it's a 1:1 reaction as it is mentioned in the question, that means the reaction will be like protein plus drug A will give us, so if we write them an equilibrium, it will be, the product will be A protein complex. Now, um, now we are given the initial concentration of the protein and the drug. Initial concentration of the protein is given as 1.5 times 10 to the power minus 6 molar, and the initial concentration of the drug is given as 2 times 10 to the power minus 6 molar. So now if we set up an ice table for this reaction right away, um, we will have to write down the concentration of the drug protein complex initially is 0, since there is no drug protein complex formed at the initial stage. And then if we consider the change as x, then the change for both of the protein and drug will be minus x, and for the drug protein complex it will be plus x. Since because it's a 1:1 reaction, that means there we don't have to raise the value of the coefficient for any of the components here. And now at the equilibrium, we will have the protein concentration as 1.5 times 10 to the power minus 6 m minus x, and then for the drug A it will be 2 times 10 to the power minus 6 m minus x, and for the drug protein complex it will be only x. Now in the question we are given that the equilibrium concentration for this drug protein complex is 1 times 10 to the power minus 6 molar, and that means the value of x is 1 times 10 to the power minus 6 molar. So we can write the value of x as 1 times 10 to the power minus 6 molar. So now if we just plug in the value of x um, in these two quantities, we can find out the equilibrium concentration of both the protein and the drug A. So we are asked to find out the value of Kc here for each of the reaction. And if we have to find out the value of Kc, we know that um, the expression of Kc will contain the equilibrium concentration of both the product and all the reactants. So we already know the equilibrium concentration of the product, but we have to know the equilibrium concentration of the reactants. And now just using this value of x, we can find the equilibrium concentration of each of the reactant. So the equilibrium concentration of the protein will be equal to 1.5 times 10 to the minus 6 minus x, and the value of x is this. So we'll write 1.5 times 10 to the power minus 6 molar minus the value of x, which is 1 times 10 to the power minus 6 molar. And from here, you can get the value of the concentration of protein with zero point 0.5 times 10 to the power minus 6 molar. So this is the equilibrium concentration for the protein. Now we'll find out the equilibrium concentration for drug A, which will be again the value of 2 times 10 to the power minus 6m minus x. So it will be 2 times 10 to the power minus 6 molar minus the value of x, which is 1 times 10 to the power minus 6. From here, we will get the value of the concentration of drug A, which is 1 times 10 to the power minus 6 m. So now we know the equilibrium concentration of the protein, we know the equilibrium concentration of the drug A, and we also know the equilibrium concentration of the drug A protein complex. So as we know all the equilibrium concentrations, now we can find the Kc value for this reaction. And as you know that the Kc value for the reaction will be the concentration of the product, that means A protein complex concentration over the reactant concentrations, which is protein concentration times the drug A concentration. And from here, the A protein complex concentration is we are given as 1 times 10 to the power minus 6 molar. So this is the value of X, and again this is um, the drug A protein complex concentration at equilibrium. Now we have calculated the protein concentration at equilibrium as 0.5 times 10 to the power 6 10 to the minus 6 molar times the drug A concentration we have calculated as 1 times 10 to the power minus 6 molar. And from here we can find the value of Kc, which will give us a value of 2.00 times 10 to the power 6. So this is the value of Kc for the protein reaction with drug A. Now in the same way we will calculate the Kc value for the reaction of protein and drug B. So we'll just set up a same reaction like this, and what we can do is we can just set that here. We'll just write in case of drug A we will have to write drug B, and the complex will be uh, B protein complex. And so the initial concentration of the protein will be same, since it is we are taking same protein concentration for both the reactions with drug A and drug B. And the drug B concentration is also same as drug A. The initial concentration is 2 times 10 to the power minus 6 molar. So the change is basically in the value of x. So the change here will be also minus x for both protein and drug. And for protein drug complex, it will be plus x. And the plus x is basically the value of the equilibrium concentration of drug B and protein complex. And this value is 1.4 times 10 to the power minus 6. So here it will be for drug B, it will be 1.4 times 10 to the power minus 6. So now again in the same way, we will find out the equilibrium concentration for protein using this quantity, and we will find out the concentration equilibrium concentration of drug B using this quantity. As we know the value of x, we can easily find this out. So the concentration of protein in the reaction of protein and drug B will be equal to 1.5 times 10 to the power minus 6 molar, which is the initial concentration of the protein minus the value of X, or the equilibrium concentration of drug protein complex, which is 1.4 times 10 to the power minus 6 molar, which is given. So this will give us a value of zero point 0.1 times 10 to the power minus 6 molar. So this is the equilibrium concentration of the protein in the reaction of protein with drug B. Then we'll find out the equilibrium concentration of drug B, which will be again the initial concentration of drug B, which is 2 times 10 to the power minus 6 molar, minus the value of X, which is 1.4 times 10 to the power minus 6 molar. From here, we'll get a value of 0.6 times 10 to the power minus 6 molar. Again, so now this is the equilibrium concentration of drug B, and this is the equilibrium concentration of protein, and this is the equilibrium concentration of the drug B protein complex. So now again we know all the equilibrium concentrations. Now we can easily find out the value of Kc for this reaction. Again, Kc is the concentration of the product over the concentration of the reactants. So the product here is drug B protein complex, and the reactants are protein. So protein concentration times the drug B concentration. And if we just plug in the values, so the B protein complex concentration at equilibrium is given as 1.4 times 10 to the power minus 6. The protein concentration at equilibrium for this reaction we have calculated as 0.1 times 10 to the power minus 6 times the drug B concentration we have calculated as 0.6 times 10 to the power minus 6, and this will give us a value of um, 2.33 times 10 to the power 7. So this is the value of Kc for the reaction of protein with drug B, and this is the value of the Kc for the reaction of protein with drug A. So from these two values we can easily see that the Kc value for the reaction of protein with drug B is higher than the value of the Kc for the reaction of protein with drug A. And as we know that the larger value of Kc means the formation of higher amount of um, drug B protein complex, and also this means that in this case the forward reaction rate is higher, much higher compared to the reverse reaction rate if we compare the reaction rate with the reaction of drug A with the protein. So the overall summary here is when the Kc value is larger, that means the reaction of protein with drug B is forming at higher rate, and we are having stronger binding between the protein and the drug B, and that means um, this drug, or the drug B, is the better choice for further research, since it has a higher value of Kc, and again higher value of Kc means more product and more faster reaction rate and stronger binding with the protein.